Hello students, myself Mrs. Vaishali from the Department of Microbiology, KTHM College. I'm here to discuss the topic of adaptive immunity, in which we will be discussing today the subtopic that is humoral immunity, in which antigen processing and presentation is the topic for today's lecture. Antigen processing and presentation is the key to immune response. Depending upon the nature of the antigen that is present, there are two different pathways by which the antigen is processed. If the antigen is an endogenous antigen, that is, that is produced within the body, then such an antigen is processed via the cytosolic pathway, and antigen is then presented to the T cytotoxic cells. If the antigen is an exogenous antigen, that is, the antigen has entered into the body from outside, like for example, a bacterial antigen, then such an antigen is processed via the endocytic pathway, and after processing, it is presented to the T helper cells. We have already studied the structure of MHC, in which we have seen that there are two different types of complexes, the MHC class 1 complex and the MHC class 2 complexes. And we have also studied in detail that the T cells are self MHC restricted, where we have seen that the T helper cells are class 2 MHC restricted, while the TC cells are class 1 MHC restricted. So, in today's lecture, we shall be studying only one type of the pathway, that is the cytosolic pathway, which is usually used for processing and presentation of the endogenous antigen. There are three different steps in this pathway. The first step of the pathway is the generation of peptides that we shall see in detail. Then the transport of all the peptides that are generated in the cytosol, and these peptides are then transferred into the endoplasmic reticulum, where they bind with the class 1 MHC molecule, and after the binding of the antigenic peptide to the class 1 MHC molecule, the class 1 MHC molecule with the antigenic peptide are expressed on the surface of the target cells. So, in this lecture, we shall be studying the endogenous pathway of antigen processing and presentation, and the three different steps involved in this pathway in detail. After the lecture, the students will be able to understand what an endogenous antigen is. They will understand how the peptides are formed in the cytosolic pathway using the proteasome that is present in the cytosol. They will also be able to understand the transport of the peptides from the cytosol into the endoplasmic reticulum, and then how the antigenic peptide binds to the class 1 MHC molecule, how class 1 MHC molecule is associated within the endoplasmic reticulum, how it binds to the antigenic peptide, and then how it is expressed on the surface of the target cell. In the immune system, there are different types of antigens that are present. Some of the antigens are referred to as the endogenous antigens, like for example, when a viral particle enters into the body, this viral particle comes from outside into the body, but when it enters inside the body, it infects the host cell, and the host cell starts producing the viral antigens. So, viral antigens that are produced by the host cells are referred to as the endogenous antigens. In addition to the virally infected cells that produce the antigens, there are tumor cells that may be generated in the body, which are the altered self cells. So, these antigens are also produced within the body, and any antigen that is produced within the body is referred to as an endogenous antigen. When an antigen enters into the body from outside, but there is no synthesis of any antigen within the host body, then such an antigen is termed as an extracellular antigen. For example, when a bacterial cell causes infection in the human body, the bacterial cell antigens are the antigens which are termed as the extracellular antigens. Because the human body does not produce the antigens of the bacterial cell, and thus they are the extracellular antigens. Now, because we have two different types of antigens, the extracellular antigens and the endogenous antigens, there are different pathways that are used for the processing of these antigens. And these pathways include the cytosolic pathway, which is used for processing the endogenous antigen. And after processing the antigen, it is bound with the class 1 MHC molecule and it is then expressed on the surface of the target cells. While if the antigen is an extracellular antigen, the extracellular antigen is processed via the endocytic pathway and then it is presented to the class 2 MHC molecule and expressed on the surface of the antigen presenting cells. We have studied that all the cells in the body, all nucleated cells in the body, express the class 1 MHC molecules. Right? And therefore, all these nucleated cells in the body can be termed as the target cells. While the class 2 MHC molecules are expressed only on the surface of the antigen presenting cells, which mainly include the B cells, the macrophages, as well as the dendritic cells. So, we'll study in detail about the cytosolic pathway used for the processing and presentation of the endogenous antigen. Within the cell, there is a small machinery that is present in the cytosol or the cytoplasm of the cell, and this machinery is referred to as the proteasome. This proteasome in a normal cell is used for turnover of the proteins that are present in the cell. For example, enzymes are present in the cells, and once the enzyme is produced, it acts on the substrate for for a limited period of time, after which there is some wear and tear that occurs in the protein, and this reduces the activity of the protein. And therefore, it is necessary that this enzyme protein should be recycled. So, such proteins that need to be recycled in a normal cell are recycled via the proteasome, where the proteasome in a normal cell cleaves the targeted proteins into short peptides, which are further converted into amino acids, and these amino acids are again reused for the synthesis of the new proteins. So, when the endogenous antigen is present, an antigenic protein is produced within the host cell. And this antigenic protein is also cleaved with the help of the proteasome. To target any particular protein for turnover or for degradation, a small protein subunit called ubiquitin is attached to the protein so that the proteasome can recognize this protein and cause the degradation of the targeted protein. This ubiquitin helps to differentiate between the targeted protein, which is targeted for degradation, and a normal protein. So, a normal protein cannot enter into the proteasome and cannot be degraded. Only when the protein is targeted by ubiquitin, it can enter into the proteasome and be degraded. This is the structure of the protein ubiquitin, which attaches to the peptide and targets the peptide to enter inside the proteasome. The structure of the proteasome is a complex structure which is made up of two subcomplexes, in which the first subcomplex is referred to as the catalytic core. This catalytic core is made up of certain small subunits which are arranged in a circular structure with a hollow in the center. The catalytic core particle has a sedimentation coefficient of approximately 20 Schwedbergs, and so it is also referred to as the 20S subunit. The second subunit or second subcomplex of the proteasome is either one or two terminal regulatory particle. This regulatory particle has a sedimentation coefficient of 19S and acts as a door or a valve which opens and closes so that none of the untargeted proteins can enter inside the proteasome. The subunits of the catalytic core are arranged in a barrel ring structure with a narrow channel in the center through which the ubiquitinated protein can enter into the proteasome so that the catalytic core particle can cleave the protein into small peptides which can be further degraded into amino acids, and these amino acids can be recycled. The proteasome requires ATP in such a way that it can degrade the antigenic peptides. The ubiquitinated protein complex enters into the proteasome, and within the proteasome, the catalytic subunit of the proteasome cleaves the peptide bonds in an ATP dependent process. So, it is an ATP dependent reaction which degrades the protein into peptides in the central hollow of the proteasome. So, this is the basic structure of the proteasome that is present inside the normal cell, which is used for the normal turnover of proteins. This proteasome is also referred to as ubiquitin proteasome. In this figure, you can see that the constitutive proteasome is the ubiquitin proteasome, which has three catalytic subunits, the beta 1, beta 2, and the beta 5 subunit. While another variant of the proteasome, which is referred to as the immunoproteasome, is the proteasome which helps in cleaving the antigenic peptides into or antigenic proteins into small peptides, and these are the variants of beta 1, beta 2, and beta 5. In the immunoproteasome, the catalytic units are beta 1 I, which represents that it is a subunit of the immunoproteasome. Beta 2I and beta 5I are the catalytic subunits whose activity is a little bit different from the activity of the constitutive proteasome or the ubiquitin proteasome. The ubiquitin proteasome has two regions, the 19S and the 20S, the 20S core. The 20S core is the central portion which forms a channel, while the 19S forms a cap which is like a valve which opens and closes and allows the entry of the ubiquitinated protein inside the proteasome. While the immunoproteasome has a variant that is present, which is referred to as the PA28 variant. This PA28 variant that is present on one end of the 20S core allows the or the antigenic proteins to enter inside the immunosome. Right? And therefore, there is a slight difference between the proteasome, which is functional in the immune conditions, as well as the proteasome that is present, that is functional under the normal conditions. The subunits of the immunosomes, beta 1, beta 2, beta 5, the catalytic subunits. The green ones are the beta 1 subunits, the red one is the beta 2 subunit, and the blue one, the dark blue one, is the beta 5 subunit. And the variants that are the immunovariants of the subunits present in the immunoproteasome. The function of these catalytic subunits is to degrade the antigenic peptides or the proteins at specific sites in the structure of the protein. The B5 subunit of the proteasome has a chymotrypsin like activity. That means it has an ability to cleave the amino acids phenylalanine, tyrosine, and tryptophan at the carboxy terminal end. That is, in short, you can say that the beta 5 subunit has an ability to cleave at the C terminal of the hydrophobic amino acids. So, the beta 5 subunit of the normal proteasome has a chymotrypsin like activity. Similarly, the beta 5I of the immunoproteasome also has a similar activity, that is the chymotrypsin like activity. The beta 1 subunit of the normal proteasome has a capsase like activity. Capsase like activity is the activity in which the protein is cleaved at an acidic amino acid residue, like, for example, the aspartate amino acid. That is, after the amino acid aspartic acid, there is a cleavage that occurs in the normal protein. But the B1 I subunit has a chymotrypsin like activity. So there is a difference between the activity of B1 and B1I. B2 and B2I, the subunits of the normal proteasome and the immunoproteasome, have a similar activity, that is the tyrosine like activity, which is the activity or the ability to cleave at the carboxy terminal end of the basic amino acids, like, for example, lysine residue and the arginine residue. So, if you see, the structure of the immunoproteasome is, is modified in such a way that more cleavage or more peptides are generated with a hydrophobic amino acid or a basic amino acid at the C terminal rather than an acidic amino acid. This enhances the binding of the peptides in the peptide binding cleft of the class 1 MHC molecule. What differences occur in the immunoproteasome occur because of the cytokine, that is interferon gamma, or the tumor necrosis factor alpha, that is produced by the T helper cells. In presence of interferon gamma or the tumor necrosis factor alpha, there are certain genes that are located in the region of class 1 MHC molecule which get activated and they encode for a low molecular weight protein referred to as LMP2 and LMP7. Two different genes get activated which produce the component LMP2 and LMP7. And these LMP2 and LMP7 are the catalytic subunits that are present in the immunoproteasome. And because of these two subunits, that is LMP2 and LMP7, there is a change which occurs in the normal proteasome and the immunoproteasome, which leads to the formation of the peptides which can bind in the peptide binding cleft of class 1 MHC molecule. So, the main function of the immunoproteasome is to facilitate the endogenous antigen presentation via the class 1 MHC molecules and expression on the surface of the antigen of the target cell. The second important function that is performed by the immunoproteasome is to preserve the protein homeostasis. Because in presence of interferon gamma, there is an oxidative stress that is induced in the cell, due to which protein degradation occurs in a larger amount. And the immunoproteasome helps to maintain the protein homeostasis within the cell. Now, the generation of peptides within the occurs within the cytosol of the target cell with the use of the immunoproteasome, which has an ability to cleave the peptides and generate large number of peptides with either hydrophobic amino acids at the carboxy terminal or basic amino acids at the carboxy terminal. These peptides that are generated have a length between 8 to 16 amino acids. Any peptides that are generated with a longer length are cleaved by enzymes that are present within the endoplasmic reticulum, that is the endoplasmic reticulum aminopeptidase enzyme, which is abbreviated as the ERAP, which cleaves the longer peptides into shorter peptides so that these peptides can be transferred from the cytosol into the endoplasmic reticulum. Now you can see that the protein is first targeted with ubiquitin. This ubiquitinated protein, it binds to the 19S regulator. The 19S regulator has a function to an important function to perform is once the ubiquitinated protein binds to the regulatory subunit, it cleaves off the ubiquitin from the protein. It opens up the alpha helices that are present in the structure of the protein and leads to the formation of a linear peptide. This linear peptide then enters through the channel of the core particle of the immunosome or the immunoproteasome, such that the wherever the hydrophobic amino acids may be present or the basic amino acids may be present, the enzymes or the catalytic subunits of the core proteasome can act on these amino acids and cleave the protein into small peptides, and the ubiquitin is recycled so that it can bind to the other proteins and target the other proteins for degradation. Once the peptides of the desired length are generated into the cytosol, it is necessary that these proteins or these small peptides enter into the lumen of the endoplasmic reticulum, because the class 1 MHC molecule is generated or is assembled within the lumen of the endoplasmic reticulum. So, the second step in the cytosolic pathway is the transport of the peptides from the cytosol into the lumen of the endoplasmic reticulum. For the transport of the proteins or the peptides from the cytosol into the endoplasmic reticulum, in the lumen of the endoplasmic reticulum, a protein which is a transmembrane protein is required, which is abbreviated as the TAP protein. This TAP is transport associated antigenic transport associated protein, which helps in transport of antigenic peptides from the cytosol into the into the lumen of the endoplasmic reticulum. This TAP protein is made up of two subunits, TAP1 and TAP2. So it is a heterogeneous protein which is a transmembrane protein, and it traverses through the endoplasmic membrane around 3 times. And each of the subunit TAP1 and TAP2 is associated with the ATP, because the transport of peptides from the cytosol into the lumen is an ATP dependent reaction. Energy is required for the transport of the peptides, and this TAP protein is specific to transport peptides within a range of 8 to 16 amino acids, and therefore the immunoproteasome is modified in such a way that the peptides, most of the peptides that are generated, have 8 to 16 amino acid residues. Any large peptides are then broken by the aminopeptidases that are present in the endoplasmic reticulum. So, specific length of the peptides, that is 8 to 16 amino acid long peptides, are allowed to be transported via the cytosol into the lumen of the endoplasmic reticulum via the TAP proteins. Once the antigenic peptide enters into the lumen of the endoplasmic reticulum, it should bind with the class 1 MHC molecule. So, the class 1 MHC molecule, the alpha chain is generated, is transported into the endoplasmic reticulum. The beta 2 microglobulin, which is a part of the class 1 MHC molecule, is also present, is also transported inside the endoplasmic, into the lumen of the endoplasmic reticulum, and the folding and assembly of the entire or active class 1 MHC molecule occurs within the endoplasmic reticulum. The alpha chain that is synthesized by the class 1 MHC genes located in the MHC molecule or the MHC gene complex is very unstable. For it to become stable, it is necessary that it should bind with a molecular chaperone, that is calnexin. When the alpha chain binds with the molecular chaperone calnexin, which is a membrane protein that is present in the endoplasmic reticulum, this calnexin, it facilitates the proper folding of the alpha chain of class 1 MHC molecule. When it folds into the proper structure, it generates the peptide binding site, and this peptide binding site should not be occupied by any of the peptide before the entire assembly of the class 1 MHC molecule. And this occurs because of a protein, that is the ERP57 protein, which binds in the peptide binding cleft of the class 1 MHC molecule, and it stabilizes the folded alpha chain of the class 1 MHC molecule. Once the ER, that is the endoplasmic reticulum P57, binds to the alpha chain of the class 1 MHC molecule along with the calnexin, the alpha chain is stabilized. Then the beta 2 microglobulin, which is not synthesized by genes of the MHC. The genes that encode for the beta 2 microglobulin lie outside the MHC molecule complex, MHC gene complex, but this beta 2 microglobulin comes and binds with the alpha chain after the folding of the alpha chain to form the entire structure of class 1 MHC molecule. This class 1 MHC molecule, that is the alpha chain bound to the beta 2 microglobulin, then leaves the calnexin, that is the molecular chaperone calnexin, and associates with the calreticulin, which is another molecular chaperone which facilitates the binding of the tapacin protein to the structure of the class 1 MHC molecule. So now the calreticulin is bound to the class 1 MHC molecule, which is a completely formed class 1 MHC molecule with the alpha chain and the beta 2 microglobulin. This calreticulin bound class 1 MHC molecule binds to tapacin. Tapacin is a protein which is associated with the TAP proteins. So it is TAP associated protein and thus called as the tapacin. The main function of this tapacin protein is to bring the TAP protein closer to the assembled class 1 MHC molecule. Right? So when the antigenic peptide enters inside the lumen of the rough endoplasmic reticulum, the antigenic peptide should not get lost in the endoplasmic reticulum. It should not be lost in the endoplasmic reticulum, and therefore the tapacin protein brings the class 1 MHC molecule close to the TAP protein, so that the peptide that enters inside the lumen of the endoplasmic reticulum can bind with the peptide binding cleft of class 1 MHC molecule, in which it dislocates the ERP57 and the antigenic peptide binds to the class 1 MHC molecule. Now, the antigenic peptide that is bound in the class 1 MHC molecule in the third step has to be transported from the lumen of the endoplasmic reticulum towards the surface of the target cell. Right? So, once the complete formation of the class 1 MHC peptide complex occurs, the peptide, the class 1 MHC molecule, it leaves the endoplasmic reticulum, and via the Golgi complex, it is transported and expressed on the surface of the antigen present of the target cell. Now let us see the overview, the entire process in the figure. The first step in the process of antigen processing is the degradation of the protein into the peptides via the immunoproteasome, where the protein enters into the proteasome, peptides are generated, and peptides of a specific size are transported from the cytosol into the uh, lumen of the endoplasmic reticulum via the TAP protein. The TAP1 and the TAP2 subunits form a channel through which the peptides enter into the lumen of the endoplasmic reticulum. When the peptide enters inside the lumen of the endoplasmic reticulum, the peptide binds with the class 1 MHC molecule, and then this class 1 MHC molecule exits from the rough endoplasmic reticulum via the Golgi network in the cell. It is transported towards the surface of the uh, target cell, where it can be presented, the antigenic peptide can be presented to the T cytotoxic cells. The T cytotoxic cells will get activated, will get converted into the cytotoxic T lymphocytes, and will help in causing the lysis of the target cell. So, this is how an endogenous antigen is processed and presented on the surface of the target cells to the to the T cytotoxic cells. Right. So, in this lecture, we have studied what an endogenous antigen is. We have studied the function of the proteasome and what are the differences between the normal proteasome and the immunoproteasome. We have also studied how peptides are generated by using the immunoproteasome, how these peptides that are formed in the cytoplasm are transported into the lumen of the endoplasmic reticulum via the TAP proteins, how the assembly of class 1 MHC molecule occurs within the lumen of the endoplasmic reticulum, taking the help of the molecular chaperones that are present in the endoplasmic reticulum. And once the complete MHC molecule is assembled, how the antigenic peptide binds to the peptide binding cleft of class 1 MHC molecule, and how the molecule, the entire molecule of class 1 MHC peptide, comes to the surface of the target cell where the antigen is presented to the T cytotoxic cell. In the next lecture, we shall be studying about the endocytic pathway, which is a pathway that is used for the degradation of the exogenous antigens, and how the antigen is processed and presented to the T helper cell.